I don't wanna grow up, I'm a curious kid. The world has 100 questions I can play with. So I'll open my arms and eyes and wonder every day till the day I die.
All right, our next speaker. Now up is UC Berkeley statistician, Dr. Stephen Bates. He'll present Being Honest with Big Data. Before coming to Cal, he's a UC Berkeley student, Stephen was an undergrad at Harvard and a grad student at Stanford, earning a PhD in statistics, I believe it was just last year. Stephen was honored with the Weiland Graduate Fellowship and the Anderson Theory of Statistics Dissertation Award. Now, as a postdoctoral researcher at Cal, Dr. Bates works with the Michael Jordan of statistics research. Indeed, UC Berkeley professor Michael Jordan. All righty, please welcome Dr. Stephen Bates.
Thanks, Tucker. So ketchup prevents cancer, okay? Or at least this is what I was told when I was reading the internet this morning. So I read this article, Saucy, Cutting Cancer to Boosting Fertility, 5 Surprising Benefits of Tomato Ketchup. And then Ed Sheeran is also selling these $1,500 bottles of ketchup. Okay. So I'll ask you, just think to yourself, so do you believe this? Are you pretty sure that ketchup is gonna, prevent you from getting cancer, or perhaps you're a little suspicious? Okay, so in fact, I have a bottle of ketchup here. I got it out of my fridge. Am I holding, world's next sort of superfood that's going to prevent me from getting cancer, among many other benefits? Or maybe you're a bit skeptical. Okay. All right, so I'm a bit skeptical. You might be a bit skeptical too. As a statistician, I think I'm trained to be skeptical, and I'm also trained to think about, why am I skeptical of this claim? Why am I not totally sure that ketchup prevents cancer, as this article might have me believe? And that's something I'll kind of unpack a little bit in today's talk about why when I see something like this, I might kind of start to, ask about how they reached this conclusion and then maybe not believe it so much. All right, now articles like this are much more common than you would think. So, I saw another example the other day. It's called Liking Curly Fries on Facebook Reveals Your High IQ. Okay, so this article, if you believe it, says the following. Facebook has this study where they looked at all of the different things people liked, and then they looked at other variables, such as in this case, they have some proxy for IQ. And they found that there is a link between liking curly fries and having a high IQ. So if you went on there and you clicked, I like curly fries, then you also are more likely to have a high IQ, perhaps, if you believe this article. Okay, so I also think that this article might be perhaps a spurious positive. But you'll see stuff like this all over the place. You open the New York Times, and you'll see something like this. Okay. Now, these are kind of silly examples, but there are much more serious examples where we encounter kind of similar facts. A really kind of important and famous one here is called Google Flu Trends. Google Flu Trends was this sort of algorithm or this product that was intended to detect early flu outbreaks based on Google searches. Okay, so Google would look at what people are searching in Google in, San Francisco or in Minnesota or wherever, and then they would say, hey, I saw a lot of searches of tissues, so I'm worried that there's a flu outbreak. Okay. Now, this was some really serious stuff. It was like heralded as kind of a big breakthrough in public health, that now we can kind of do this digital detection of things like the flu. This is great. This can tell us exactly when there's going to be flu outbreaks. We can send people, flu vaccines, or we can send extra help if they need them, and so on. However, in the years, this came out in 2009. In the years between 2009 and 2015, they realized that this is not working at all, that they didn't have any predictive power, that all these kind of patterns that they thought they had detected in the data were kind of spurious and weren't really there. And the whole product came to a close in 2015, and it's kind of widely now panned as a huge failure. So Google Flu Project shows the failings of big data, this whole kind of subfield of public health, this kind of like digital epidemiology it was called. It kind of was very excited about this, and now it their kind of flagship product had broken down. Okay. So this was a huge failing. It was very expensive and very harmful. You know, people were literally allocating public health resources based on this algorithm, and it turned out to not work at all. You know, 5 years later, we found out it didn't work at all. And perhaps, you know, even more scary, that this article sort of indicts all of big data. You know, so as a statistician, as a scientist, I think there's a lot of value out there that we can get. We can learn a lot about the world from data, but there's articles just kind of saying, oh, you know, maybe we're always going to fail like this. We can't trust this at all. Okay. So I'm very worried about this. And, you know, this is one of these types of things are what led me to be a statistician. And one thing I'll talk about today is kind of how to be honest with big data like this, or how to not fool ourselves, not just fooling other people, but fooling ourselves when you have data like this. How can I really draw conclusions that I can, you know, be 100% confident in? All right. How can I do that? All right. So the underlying problem, or kind of an underlying problem in these examples that I've showed you, is something, there's a technical name for it, but for now I'm going to call it the jelly bean problem. And the reason I'll tell you a story about why it's the jelly bean problem. Okay. All right. So my friend came to me and told me jelly beans cause acne. Okay. Jelly beans cause acne. That doesn't sound good. I don't want acne. Maybe I should cut off jelly beans, you know? Okay, but I'm a scientifically minded guy. So I said, okay, scientists, let's go do a study. Let's see if jelly beans cause acne. Okay. So the scientists go and do a study. They check out jelly beans. They see if they cause acne or not. And they come back and they say, we found no link between jelly beans and acne. And I said, okay, that's great. I can go back to eating my jelly beans. You know, life is good. But then my friend says like, no, that's not right. It's only a certain color of jelly bean that causes acne. It's, you know, and I said, okay, scientists, can you check this out? Can you check out if there's any one of these colors of jelly beans that causes acne? All right, and it turns out there's 20 colors of jelly beans, and the scientists are going to check them out. So they start with color number 1. And they said, we found no link between purple jelly beans and acne. I said, okay, great, sounds good. We found no link between brown jelly beans and acne. Okay, great, that also sounds good, no link. We found no link between pink jelly beans and acne. Okay, great. No link between pink jelly beans. And so on and so on and so on and so on. No links so far. And then one day they come back and they say, we found a link between green jelly beans and acne. Okay. We found a link. There's a link. And I said, oh no, there's a link between green jelly beans and acne. Okay. But then they finished the study, nobody else, just the green jelly beans. All right. And then the headline on the news is, green jelly beans linked to acne. Oh no, we should not eat our green jelly beans. Okay, so what happened here? And the newspaper even reported some statistical measurements. They said that they're 95% confident that the green jelly beans, cause acne. All right, so what happened here? And I'm gonna tell you that I'm a skeptic and I don't think the green jelly beans cause acne. So what happened here is 95% confidence means I was only 5%. I only have a 5% chance of making a mistake, right? In any one instance, I am only going to make a mistake 5% of the time. Okay, so the chance that I made a mistake here was only 5%, here 5%, here 5%, and so on. But I did this 1, 2, 3, 20 times. Okay, so I only had a 5% of chance of making a mistake, but I did it 20 times. So what's the chance that I made any mistakes? Okay, it's not that low, right? I'm pretty likely to make at least one mistake if I do this 20 times. So that's what's happening here. The green jelly beans just appeared because I was unlikely to make a mistake, but I did it 20 times. When I do it 20 times, I become pretty likely to make a mistake, and I really can't trust this conclusion. Okay. All right, so this is the jelly beans problem, and this is what's, also it's the problem with ketchup. It's the problem with curly fries and IQ. It's the problem with certain words predicting the flu. Like all of it is kind of this thing. It's like if you look too hard, you're going to fool yourself. Okay, so you need to account for that. You need to think about how hard did I look, and I need to correct for that. All right, now this is not just a silly problem. This is kind of at the heart of many modern scientific investigations. One problem I work on and I care a lot about is genetic studies, so genome-wide association studies, we might call them. In early 2000, the Human Genome Project was completed, and this was one of the major scientific accomplishments of the last 25 years, perhaps the single most important scientific accomplishment of the last 25 years. So they finally took a kind of human genome, they were able to sequence the entire thing and look at it from start to finish. And we had one human that we measured. Now, since then, there's been an explosion of measurement technologies, and we've measured, hundreds of millions, we've sequenced, or at least partially hundreds of millions of people, many different species of plants, animals, and so on. So we have this huge amount of genetic information that we can now use to understand biology, and then downstream, do things like predict medical outcomes or kind of build medical treatments. All right. So again, here's a picture of Francis Collins receiving the Presidential Medal of Freedom from George Bush at the time. Again, one of the most important scientific undertakings of the last 25 years. Now, I work a little bit on these problems subsequently. I'm going to call them genome-wide association studies. The point here is I get a bunch of genetic information and I want to understand something about, let's say, a health outcome. Okay, so perhaps, for example, one thing I do study, but just as an example also, heart disease. So I want to know what genetic factors affect heart disease. So how does one study a question like this? Well, you go and you collect the genetic information from a bunch of people, and then you look at the people who say have heart disease, and you're going to compare them to the people with no heart disease, and you're going to see, are there kind of specific, I'll call them genes. Are there genes where there's a difference between the people who have heart disease and the people who do not have heart disease? Okay. So in this example, just look at this little region here that's sometimes red. I'm going to look at that region of the genome, say, and I'm just going to count how often does a person have the red version versus how often do they have the normal version. There's a red version and there's a normal version, and everybody has one or the other. Okay, so suppose in the heart disease population, in this case, 58% of people had the red version. In the no heart disease population, 17% of people had the red version. So in this case, I would say these things are not really the same thing. And I would say, okay, maybe the red version here has something to do with heart disease. Okay, so that's big picture how these studies work. I have people with heart disease, I have people without heart disease. I look at some region, say this red region here, and I just compare the two. Okay. But now I don't just do this for the red region. I do this for the entire genome. I do it everywhere I possibly can. Okay, so I start, let's say, at this chunk of the genome and I measure it. And I say, is this different between people who have heart disease and people who do not have heart disease? I move to the next one. I do it here. I say, is it different for people with heart disease, people without heart disease? I go to the next one. I ask the same question. Is it different for people with heart disease, without heart disease? And so on and so forth across the entire genome. Okay, I do it over and over and over and over and over, and I look at all of these different genes. Or you can even go finer scale than genes. You can look at very small regions of the gene. All right, so if you follow a little bit what I'm saying, what happened here is that, let's say I did this 10 million times. I now have 10 million jelly bean colors, right? I have 10 million different little windows where scientists went off and said, did gene 1 affect heart disease or acne or anything like this? Okay, so I have 10 million jelly bean colors. It's the jelly bean problem on steroids, and there's a technical name for this. It's called the multiple comparisons problem. So multiple comparisons is the term of art that statisticians, geneticists, and other scientists would use to discuss problems like this. Okay. So how would I analyze data like this? I can't kind of just do the thing I was doing when I only had one at a time. I need to be a little bit more careful about it. And we've designed a lot of sort of statistical algorithms or ways of looking at data like this and trying to get correct answers from it. So I'll tell you just a little bit about that. Before I talk about the problem of 10 million jelly beans, I'll just, again, kind of refresh our memory on what do I do when I only have a single gene. Okay. So this is what I was describing before. How do I know if something is true? How do I know if this red region is actually causing heart disease or no heart disease? Is it actually affecting it? So what we do in statistics is we try to very precisely explain the amount of evidence that we have in the data. Okay, so let's just, again, look at this one gene and let's talk about how I would analyze this. Let's suppose I saw a data set that was the following. I had 70 people out of 120 that had the red version in the heart disease population. Okay, and maybe I had 20 people out of 120 who had the red version in the no heart disease population. Okay, Now, in this case, I think we would all kind of agree, there's a huge gap between 20 and 70. I'm pretty sure these are not the same, okay? So in this case, they're definitely not the same. The heart disease is not the same as no heart disease. And so it looks like this gene is important. Okay, again, the flow of logic here is, if I can tell that there's a difference between these two groups of people, that means something interesting is happening. That means the gene is potentially important. Okay. So when I see 70 out of 120 and 20 out of 120, they're definitely not the same. You can kind of intuitively kind of guess that. Okay, what if I saw something a little bit different? What if I saw a bit less of an extreme data set where 35 out of 120 people had the red version in the heart disease population, and 20 out of 120 had the red version in the no heart disease? Okay, so 30% and 17%. And now I would say, okay, is there a difference between these two groups? Like, do I think that this red version actually has anything to do with heart disease? Or did I just get a little unlucky and I saw kind of a unbalanced sample? Well, you can do the math. Statisticians like me, we do the math to kind of precisely understand how much evidence there is in this case. In this case, they're probably not the same. So it's probably these two groups of people are different. They have different frequencies of the red version. Okay, but it's kind of possible that they're the same. They're not so extreme. So in this case, I would say I'm only moderately sure that these groups are different, and therefore I'm only moderately sure that kind of this red version of the gene, has anything to do with heart disease. Okay, so this is what we do in statistics. We try to kind of precisely state how certain or not we are about these outcomes. All right, and then lastly, if I saw an even less extreme data set, if I saw 25 out of 120 had the red version here, 20 out of 120 had the red version here. Here I would say, I think it's very plausible to me that this red gene has nothing to do with heart disease, that these are just equal. Probably there's no difference here. If there's a difference, it's pretty small. So if I saw a data set like this, then I really can't conclude anything is going on. Okay, so this is what I'm doing when I see one gene. This is just kind of how we do statistical reasoning in this case. Is we just say, is it plausible that the red gene is not doing anything and that they're equal, or is that implausible? Is that kind of very unrealistic given what I've seen? And we could be anywhere in between. Now remember we were talking about the jelly beans problem. This is not the jelly beans problem. This is just basic kind of one at a time statistics. What happens in the jelly beans problem? Okay, what about when I have 10 million sites? I measure your entire DNA. All right, we're going to need some kind of more subtle statistical reasoning. We'll need to do something a little more advanced than just kind of a yes or no decision or something about every single one. We're going to have to think about everybody at the same time. Okay, and the kind of technical term here is the false discovery rate. We're going to think about what happens when I make some big batch of statistical claims at the same time. Okay, so to talk about what the false discovery rate is, I'll give you just a very quick outline for the flavor of it. Okay, suppose I had one site. Site here means gene, one site on the genome. And I had a 0.1% chance of a false positive. So this 0.1% is usually a number I get to set in advance. The genetic researcher or the statistician gets to set this in advance. So suppose there's a 0.1% chance of a false positive. Okay, so I did my test where I have a pretty low false positive rate. Okay, but suppose I did this 10 million times. There's 10 million sites on the genome. How many false positives do you expect to have? Well, I expect to have about 10,000 false positives, right? 0.1% of 10 million is 10,000. So if I did this individually for all these 10 million things, I know I'm going to have about 10,000 false positives. Okay, 10,000 false positives sounds bad, but it's only bad in context, right? In these particular studies, I might find a lot of true positives. I might find an overwhelming number of true positives. And it's not unrealistic that I would see something like 50,000 true positives. So I might find 50,000 different regions where it looks like something promising is going on. Okay. Now, if I see this, if I saw 50,000 detections, and I know that I expect about 10,000 false positives, what does that mean? Well, it means that about 20% of my discoveries are false, right? I know about 10,000 of them are false. I saw 50,000 things total. The false discovery rate, which is what we would call this, is 20%. Okay. And 20% is really not that bad, right? Like if I hand the doctor this list of things and I say, most of these are true, in fact, 80% of them are true and 20% of them are false, that's reasonable information depending on the context. That might be kind of a reasonable notion of certainty for the doctor in that case. All right, so in this case, we would report to the doctor perhaps these 50,000 sites, and we can tell them that 20% of them are wrong. I can't tell you which are which. It's too hard, but I know that about 80% of these are correct. Okay. So this is called, again, the false discovery rate. The change in paradigm here is that instead of kind of looking at one at a time, I'm now combining all, 10 million tests or 10 million kind of different pieces of information into one batch, and then analyzing the error rate of that whole batch. Now, this is a pretty active topic in statistics about how you would achieve something like this. So here, everything just worked out that I got a 20% false discovery rate, but there's a variety of algorithms that will kind of do this for you automatically. So if I ask, say, for a 10% false discovery rate, well, I know I can't return all 50,000 of these, because if I return all 50,000 of these, my error rate is 20%. But maybe if I take the most promising ones of these 50,000, the most promising 20,000, say, maybe I could achieve 10% and so on. So there's algorithms that will do this for you. You kind of put all this information and then it will tell you your level of certainty across a variety of different thresholds. Okay. All right. So I'll wrap up here. So what I showed you today is kind of this jelly beans problem. The reason it's coming about in our modern age in 2021 is that we have these massive data collections. I can just measure everything, for example, genetics or clicks on curly fries or ketchup and mustard and whatever that you happen to eat. Then what I do is I put it in my computer, and your computer can do millions of things. Your computer is like really fast. It can try, gene 1, it can try gene 2, it can try gene 3, it can try ketchup, it can try mustard and mayonnaise, it can try every possible combination. But what that means is you have millions of jelly bean colors, and that you have tried millions of different things. And that if you want to draw real conclusions from that, you have to kind of be honest with yourself that you tried 10 million jelly bean colors. Okay, I'll leave you with this quote by some famous statisticians. Modern science faces the problem of the selection of many promising findings from the noisy estimates of many. Thank you very much for your attention. I'd like to take questions you have about big data and being honest with it.
Stephen, thank you. No question has, all right, American Sign Language. No question has popped up in the chat yet, so allow me to ask a couple questions. First, I must know if you happen, well, I've got to say how much I love curly fries, so please, a little round of applause for me for that. That Google flu study, big data analysis that you mentioned, lasted for how long?
About 6 years.
And it ended up providing no insights to the arrival of a flu epidemic at all?
That's right. It turned out to be all wrong, basically. So like they were never able to make accurate predictions into the future. What happened was they had found a lot of these little relationships that appeared really promising, but then they all kind of turned out to be false positives, or they were all transient and they went away. So it turned out that the predictions were based on kind of silly things like searches for high school basketball, which is correlated with season of the year. So it carries a little bit of prediction about flu seasons, but nothing more than you knew by knowing the date anyway. And then after a while, they found out that they had no predictive accuracy.
Huh. I see that Peter M's question seems to focus on this too. Just allow me to pursue this. It just seems so reasonable that during the time it would take to do large-scale testing of the arising of flu, and let's say in the fall, that one could look at Google searches to determine whether there's a surge in purchase of Kleenex or whatever it is. But it didn't pan out. That just astounds me. Yeah.
Yes, I see Peter's question here. Why did it take so long for them to figure it out? Yeah, I don't know the question to this. I think it's, I think it's just a question of sort of, it does seem extremely reasonable, right? Like as Tucker said, if you told me that you could predict the flu based on various searches, I find that kind of plausible. I guess they just, over time they continued to try to validate it, and it didn't work. So
I want to note that we have two experts in the audience. We have Paul Switzer from Stanford, and we have Jacob Bien from USC here, who may ask very hard questions. I'm not going to go them into it, but I see here Dave has asked, are there any other solutions to the multiple comparisons problem besides stating a false discovery rate?
Yeah, that's a great question. So there are, and I should say here I've kind of set it up where I just stated the false discovery rate, but you could do it a little bit more clever where I ask in advance for a 5% false discovery rate, and it will kind of only find things that have a 5% false discovery rate. There are other approaches. If you're familiar with statistics, there's kind of a parallel track of statistics called Bayesian statistics that has techniques for dealing with things like the multiple comparisons problem. There's also, kind of, I'd say more straightforward techniques, which is just maybe you might manually only look at 2 or 3 different things. You might pre-specify, I'm only going to look at a few different things, and so on. But yeah, there are other techniques. I'd say that the main other one would be kind of Bayesian statistics, which has this nice feature that in some sense it corrects for this automatically. But I can't do much more than give you that little teaser.
Mm-hmm. Stephen, allow me one more question. I would imagine that a deep understanding of statistics is beneficial to many corporations. That is, one's job hunting prospects are raised by one's knowledge of this kind of statistics. Is there any road to that kind of deep statistical understanding besides the study of mathematics? Could somebody, for instance, a psychology major, an English major, a non-math whiz, find success in data analysis in statistics?
Oh yeah, that's a great question. Yeah, I would say more and more statistics is, I would say, kind of diverging from mathematics, not in a bad way, just in a way that the field is more mature. I would say that a lot of people who are really interested in something like designing methods for genetics really love math, but there's a lot of problems in statistics that are not about math. They're about more conceptual things, like I showed you today with the jelly bean problem. And so there's a lot of room, I think, for kind of being a really careful statistician without liking math, without really kind of, doing that for its own sake, just because you're really interested in drawing the right conclusions in something like, like psychology.
Thanks, Stephen. Dave asked the question, is p-value hacking another name for the multiple comparisons problem, or is that something else?
Yeah, so p-hacking is a similar phenomena where the multiple comparisons happens kind of on purpose or because of an active researcher. So p-value hacking, it is the multiple comparisons problem in a certain sense, but what happens is it's, I tried jelly bean color 1 and it wasn't less than 0.05. So then I say, oh no, I need to publish my paper. So I said, oh, you know what? I'll try a different color jelly bean. I'll try jelly bean color number, I'll try blue this time. And then if blue doesn't work, then I say, oh no, I still need to publish my paper. Let me try green. And then I keep doing it, and I keep doing it, and I keep doing it. So they're extremely related. It's just the p-hacking, we would kind of emphasize the part where it was due to a person, continuing to try really hard to find that that significant result to, say make a really nice headline about curly fries or something.
Great.
It's actually a little more invidious than that. Hi, Stephen. In that there's something called the file drawer problem, which also relates to p-hacking, in which case the researchers may not even report all the colors of jelly beans that they tried. So it looks like they don't have it looks like they may not have that problem. That's simply they withheld a lot of the investigations they did. And basically they were not distinguishing between what might be considered an exploratory study versus one which is a more properly called confirmatory study. I'd just like to put those 2 cents in there.
Thank you, Professor Switzer. Thank you. All right. I want to thank everybody for coming up to their computers tonight.